Hi, everyone. Thanks for joining the Boston Epigenetics Society and our March webinar. I'm just going to give a couple minutes to people and give people an opportunity to log in. So if you could just hang tight, we will get started in another minute or so. Thanks everyone for joining the Boston Epigenetics Society. We're very excited for our March webinar. I know people are still logging in, but I want to give as much time for the important stuff, the actual webinar, as possible. So I'm going to get started now. Thank you for joining us. If you're not familiar with the Boston Epigenetics Society, our mission is to promote epigenetics and epitranscriptomics research by fostering connections between academia and industry and pulling people together. And we do that by hosting webinars. And I want to really thank Cantata Bio and Active Motif, especially for hosting these webinars. And our sponsors include Cantata and Active Motif, as well as 10x Genomics and Biomedical. We also are very fortunate to have a great leadership team. And I have the names of the folks over here to the left that help us out with all of the planning and organizing. And we're looking to expand, and we'd like to have more events in the future. To have more events, it helps to have more people to help share the work. So if you're passionate about epigenetics and you'd like to become more involved with the society, by all means, please reach out. We're thinking about doing an in-person event and doing in-person events of like a whole day symposium needs lots of help and support. So if this is something you'd be interested in, please reach out to one of the folks on the leadership team or email us at bostonesociety@gmail.com. Another way to keep in touch with us is right now, the best way to follow us is on our LinkedIn page. So you can search for the Boston Epigenetics Society on LinkedIn, or you can email us. And hopefully coming soon will be a website that we're working on. In terms of upcoming events, if you'd like to come see us in person, we're going to be at the Festival of Genomics and Biodata at the BCEC on June 12th and 13th. We're still getting the details together for our next webinar, so please stay tuned for that. And our next in-person networking event will be focused around the Festival of Genomics. So we know that'll be June 12th. The details are still being ironed out. Stay tuned to our LinkedIn page for more details there. So without further ado, I'm going to turn it over to the MC for today's events, John McShane from Biomedical. And before I turn it over to him, I just want to remind everyone that at the bottom, you'll see there's a chat window and a Q&A window. The chat window is for any sort of technical questions that you have regarding the specific webinar, but all questions for the speaker should be directed to the Q&A portion. So as questions come up during today's webinar, please direct them to the Q&A section. And at the conclusion of today's webinar, John will be asking Rong those questions. So without further ado, let me please turn it over to John. Thank you very much.
Hello, everyone. I'm John McShane. I'm the Director of Business Development for Biomedical. It's my pleasure to introduce Rong Fan. Dr. Rong Fan is the Harold Hodgkinson Professor of Biomedical Engineering at Yale University and Professor of Pathology at Yale School of Medicine. He received his Ph.D. in chemistry from the University of California at Berkeley and completed postdoctoral training at California Institute of Technology before joining the faculty at Yale in 2010. His current interest is focused on developing microtechnologies for single-cell and spatial omics profiling to interrogate functional cellular heterogeneity and intercellular signaling network in human health and disease. He co-founded Isoplexis, Singleron Biotechnologies, and Atlas Omics. He served on the Scientific Advisory Board of Biotechne. He is the recipient of numerous awards, including the National Cancer Institute's Howard Temin Career Transition Award, the NSF Career Award, and the Packard Fellowship for Science and Engineering. He has been elected to the American Institute for Biomedical and Biological Engineering, the Connecticut Academy of Science and Engineering, and the National Academy of Inventors. So without further ado, it's my pleasure to introduce Dr. Rong Fan.
Yeah, thank you, John, and thank you, Brian.
I found this on the web, for he served on the science.
Okay, can you hear me now? Okay, great. Yeah, thanks, John, for the very kind introduction, and Brian and the whole community of Boston Epigenetic Society for having me. I'm gonna just dive in to my talk today, which is the spatial epigenome sequencing to map human tissue development, aging, and diseases. And this is my disclaimer. And I want to start with very early on my own experience in the field of single cell and spatial omics. 2006, when I switched my career from physical science to biomedical research, literally I knew nothing about biomedical research at all. I have to raise something about cancer biology. This is a review article written by Professor Joanna Joyce when she was still at MSK. And I learned two things, right? So the tumor microenvironment extremely complex. If you really want to understand the biology of the tumor microenvironment, you need to have better tools that can resolve this heterogeneity and the single cell resolution. Number two, ideally, you can resolve that in a spatial tissue context, because the cells are not sitting in the tumor microenvironment in completely random fashion. They form very complex, intricate spatial tissue architecture. But unfortunately, at that time, I call that the ice age of single cell biology or single cell omics. If you want to look for the tools to look at even just a dozen proteins on a surface of the cell secreted by the cells, there are literally no tools available to allow you to do that. I was very interested in a protein secreted by single cells to mediate the cell-cell interaction, cell-cell communication. That was the first technology I proposed to develop, and eventually we published a number of papers to report this single cell high-plex protein secretion technology, but also demonstrate some very unique applications in both cancer and immunology research. We continue to use this technology in conjunction with the latest breakthroughs in single cell sequencing to investigate how T cells actually work, and in particular the engineered T cells like CAR T cells, and how does that sort of function of T cells in an infusion product, for example, in the CAR T clinical trials, can predict the long-term response. We're still working on that, but as I said in the beginning, that addresses only one of the two questions I discussed, like a single cell resolution, but this does not give you any spatial information. So people realize that challenge, but also realize that importance to have spatial resolve omics mapping very early on, about like 10 years ago. And many different technologies emerged to address this challenge. Broadly speaking, there are two different directions to develop spatial omics technologies. One is based on imaging. If you can image over and over from just 2 or 3 proteins to hundreds, and all the using fish to image up to the entire transcriptome, and that's absolutely a major leap. It's not just an incremental sort of improvement of the imaging technologies that give you sort of the genome-wide, protein-wide information. And the second direction is a little bit kind of newer direction. Instead of imaging over and over, and you can use high throughput NGS sequencing to somehow reconstruct the spatial omics information, either transcriptome or protein. But as Brian was kind of asking if you are passionate about epigenetics, this is the community. I'm passionate about epigenetics very early on. That's why when I developed this technology, I was excited about the potential to really unlock spatially resolved epigenomics. This approach sort of uses NGS-based high-throughput sequencing to map omics in a spatial tissue context. Start with this landmark paper published by Joachim Lomberg in Sweden to use the DNA marker array to capture messenger RNAs released from the tissue section you place on the glass slide. And since then, there's many different technologies that have been reported, but follow pretty much the same fundamental principle coined by Professor Joachim Lomberg. That's the Barker solid surface for spatial for messenger RNA capture and the spatial transcriptome sequencing. But what we came up with many years ago, about 5 or 7 years ago, was totally opposite. Instead of capturing transcripts or whatever biomolecules to the surface of the Barker surface, we send those DNA barcodes into the tissue, and turns out this is a much more versatile way. Demonstrate for the first time spatial multi-omics, transcriptome, and dozens of proteins. And later, as I said, we are passionate about epigenetics, and largely because that biologically is much more fascinating. You understand not just what genes are expressed, but actually the mechanisms controlling the expression of those genes, and eventually determine why that cell is that particular cell, not in a different cell type, right? Okay, when we published this paper, kind of 2020 in Cell, and we demonstrate this in-tissue barcoding using the microfluidics cross-flow barcoding approach, but in theory, you can also come up with other methods to do in-tissue barcoding, but the fundamental principle here is totally different from the sort of the Barker surface for biomolecular capture. And so after we published this paper a couple of months later, and Nature Method choose the spatial transcriptomics as the method year, they also highlight our paper saying the multi-omics sequencing goes spatial now. I was pretty happy with that. So multi-omics going spatial now, but I was asking my postdoc, okay, that time we demonstrate dozens of proteins, can we go to hundreds or thousands? Because to my standard, I think dozens of proteins may not really represent kind of proteome scale analysis. Turns out that worked out very well, and we're able to incorporate 300 DNA antibody conjugates with the whole transcriptome sequencing to perform the spatial multiome profiling, and it works in for a variety of different tissue types, including mouse and a human. This has been published last year. I'm not going to talk about the details. Okay, the reason why I'm here is, of course, beyond the spatial transcriptome and the panel proteins, what we can do further in the spatial omics field. And very early on, I was not trying to develop another spatial transcriptome sequencing technology at all. And I was much more excited about the mechanism and epigenetic level regulating gene expression, and there are many different gene regulation epigenetic mechanisms out there, right? And the specific histone modifications, DNA methylation, and a 3D genome, and also global chromatin accessibility, and so on and so forth. In theory, you can do all of those using our in-tissue barcoding approach, and we decided to start with these two, okay, global chromatin accessibility and specific histone modification analysis. For global histone chromatin accessibility analysis, there's a very elegant chemistry called a taxic developed by Howard Chang and Will Greenleaf at Stanford. And so basically use TM5 transposes to segment genome DNA and open chromatin loci and insert the DNA oligo you pre-assemble with the TM5 transposes into the open chromatin region. And then when you sequence the genome DNA, you can see where that DNA oligo you insert into the genome DNA, and then you can map down where the open chromatin regions are. So in most cases, when you do either bulk or single cell ataxic, people always tell you, okay, isolate high quality nuclei, that's most important. But we found no, totally unnecessary. If you have very thin tissue section, you can just go ahead to do TM5 transposition to insert the DNA oligos into the open chromatin region of the genome in every single cell, every spot. But afterwards, we use this cross-flow microfluidic cross-flow barcoding to ligate barcodes A and B together, creating this 2D grid of tissue pixels. Every pixel has unique combination of A and B to determine the spatial address code for every single read you're gonna get out of your sequencer. As shown here, we have the data from the embryonic mouse liver, pixel by pixel across the entire embryonic mouse liver. You can see those individual data traces, but you can also aggregate them to produce the pseudo bulk ATAC data. That turns out to be in very strong concordance with the ENCODE reference data. But if you really look at the signal to noise ratio, seems like we have even better signal to noise ratio. So from all the spots, all the pixels we sequenced in the spatial ataxic, you can take every pixel to perform this unsupervised clustering, very much like how you do a single cell ataxic data clustering analysis. And then you'll see different clusters right here. But what is different from your single cell ataxic is you know exactly where every single pixel is in a tissue. So now you can put them back to the tissue and see whether or not the different clusters correspond to different tissue types or even the whole organ. For example, right here, you'll see the liver. It does correspond to liver right here. So in the brain region, you already see quite a few different clusters associated with different neuronal cell types that have started to develop at the stage of the mouse embryo E12. And this is a very small cluster, this yellowish right here. And we were not able to find sort of the reference data for this cluster, but when we put it back to the tissue, it's, oh, that's in the spine. It's very clear. So now you can see sort of the spatial resolved chromatin accessibility can very nicely map the different tissue and cell types directly in a mouse embryo. But you can also visualize individual genes as showing here for like a 6 selected genes. But keep in mind, you are not looking at gene expression. What you are looking at is in TSS enrichment score. Basically, you are looking at an epigenetic state rather than gene expression. So we did a kind of comparison or benchmarking against the encoded reference data across all different tissue types, major tissue types right here, kind of liver and a limb and a different kind of brain regions. We do see pretty good concordance between our data and the ENCODE reference data, but our data you see much more granular because we really map down spot by spot, pixel by pixel, rather than physically dissecting the entire organ to perform ataxic, which wasn't the case in the ENCODE reference data. And so what is much more powerful we always try to do is, and if you can integrate the single cell sequencing data and spatial ATAC or spatial other type of spatial omics data to perform this co-embedding. And then you can transfer the cell types identified by single cell sequencing to the spatial spots, the spatial pixels. And so this is the single cell RNA sequencing, single cell transcriptome sequencing data, and the whole data set is well annotated, you know, all the different cell lineages at the stage of the mouse embryo E12 to E13-ish. So now when we do the co-embedding, you see where the different black dots, so the pixels from our spatial ataxic, co-localized with different clusters or different cell types in this case. And if you see those spots, for example, co-localized in this kind of green cell type, you know, okay, that's the dominant cell type in those tissue pixels. So now you can do this sort of label transfer and then go back to visualize where those different cell types are. And just to show you a couple of those, for example, definitive urethral lineage cells and the spatial red blood cells at this stage are actually produced in liver, not in bone marrow. And so now you do see they are enriched in liver. You already see a lot of different neuronal cell types like excitatory neurons and post-mitotic premature neurons and some oligodendrocyte progenitors. Now you can see where they are in a tissue-directed visualize their spatial localization. But again, I want to emphasize that the purpose here is not to find where the different cells are, but to identify where the epigenetic state of the different cells and visualize the epigenetic state in a tissue. We also perform this type of spatial chromatin accessibility analysis in the postnatal mouse brain, for example, in this region, which is actually very complex. And after we perform the spatial ataxic analysis and clustering, 7 major clusters identified, and then you can go back to see where the different clusters are. So now you see the clusters correspond very well to the different regions of the brain, part of the inner cortical layer and the corpus callosum and the striatum and so on and so forth. And in particular, I think the striatum is a relatively thin, corpus callosum is a relatively thin layer. You can resolve very well, but many other kind of oligodendrocytes also migrate to other regions of the brain. You also see those kind of yellow spots in other parts of the brain as well. Similarly, you can visualize individual genes associated with different tissue regions and the brain regions. But keep in mind, what we are looking at is not just a gene expression, it's really what the epigenetic state of those genes. So we have been very interested in immunology and those lymphoid organs for many years, and we decided to apply the spatial ATAC to map the human germinal center reaction in the lymphoid tissue, in this case, in the tonsil. And we decided not to do very detailed, kind of very high resolution clustering, just to try to identify major clusters first. And the major clusters turned out to be like a follicular and extrafollicular and the crypt, which is perfect. Yeah, it's exactly what we expected. But in this case, you can look at all the individual markers, including canonic surface markers, but also the master transcription factors associated with different cell types and in different regions. For example, the PAX5 is the master transcription factor for germinal center B cells, but also expressed in sort of other cell types as well. As you can see, they are strongly enriched, entities strongly enriched in the germinal center. And so BCL6 is a master transcription factor for follicular helper T cells, but also expressed in the germinal center B cells. It's exclusive accessible in the germinal centers in the follicles. And interestingly, we saw a lot of FOXP3 signal in the extrafollicular zone, and we still don't fully understand why, but we did experiments to look at protein expression and the gene expression of FOXP3. We don't really see a lot, and we know FOXP3 is a regulatory T cell transcription factor, and so I think that's interesting. I think the hypothesis is you have a lot of cells in the extrafollicular zone. They are epigenetically accessible for FOXP3, and they may have some potential to become a regulatory T cells, although at this stage they're not ready. So this is totally different message you get out of this spatial epigenome analysis as compared to sort of the spatial gene expression or spatial protein analysis. You do see the epigenetic potential or the epigenetic sort of the readiness for those cells eventually, if necessary, they can turn themselves into regulatory phenotype. Again, so combining single cell sequencing data and spatial ATAC, you can identify different cell types and visualize where they are. For example, naive B cells to germinal center B cells and some macrophages surrounding the vasculature region. And but what is much more exciting is you can really look at the dynamics of the cell differentiation from naive B cells to germinal center B cells. Similar to single cell sequencing data analysis, you can perform this pseudo time and on genes associated with this pseudo time analysis all the way from naive to germinal center B cells. But what is different here is you can directly visualize this differentiation process in the tissue. Say, oh, the naive cells, the bluish cells, where are those cells, and those kind of yellowish cells more towards GC B cell phenotype, where are those cells? You can directly visualize them in the tissue. So recently, we are excited about applying this spatial epigenome sequencing to human cancer, for example, glioblastoma multiforme. And this is a beautiful work from Mario Suva's lab at MGH, delineating the transcriptional phenotypes of human glioblastoma. They identify 4 major subtypes, and 3 of them are dominant. And so now we're asking whether or not those different transcription phenotypes also can be resolved at an epigenetic level. And so we perform the spatial ATAC to map, I think we have data from 10 to 12 different human glioblastoma tumors now, but just to show you one of those. And so this is a piece of GBM tumor tissue section, and we have very good sort of the total number of reads and the TSS score from the sample. After integration with the single cell reference data provided by Dr. Mauro Suva's lab. And so now we can identify all the major cell types. Malignant cells are the tumor cells, but you can also see many other cell types like the tumor-associated macrophage, and this malignant actually just, and within this malignant cells, we can further identify the different subtypes. It turns out in this tumor, majority of the tumor cells are NPC-like, okay, based on the transcription phenotype. But we can also see other cells, other subtypes, and even some of those are relatively sparse, like the AC-like. So from the same tumor, we can see all 3 different major subtypes, but of course, one of those is dominant, but you can now directly visualize other subtypes directly in the tissue. You can further look at gene activity scores in this human brain tumor samples for all the different genes you are interested in. You can also look at different motifs. So I think that's a very exciting. The first time you can directly visualize the epigenetic state, and the epigenetic states I think do correspond well to the transcriptional heterogeneity identified by Dr. Mario Fuva. And so that's global accessibility of the chromatin state in the tissue using spatial attack. And now we are asking, can you further look at the specific histone modifications as well? And that's pretty straightforward based on similar chemistry using TM5 transposition, but you use the antibody to recognize the specific histone modification first, and then the protein A tether TM5 transposes to bind to the specific antibody against the different histone marks, and then you activate the TM5. You can cleave the genome DNA and the sites sort of associated with different histone marks, associated with different histone modifications. So that chemistry is called a cut tag, developed by Professor Steve Hinikoff at Fred Hutch Cancer Center. So we apply this to 3 different histone marks here: K3, K27 trimethyl, K4 trimethyl, and K27 acetylation. So we found even without doing multiplex cut and tag, we were able to kind of resolve how different histone modifications control the tissue development in this mouse embryo in a sort of differential manner or synergistic manner, or kind of sometimes called a multivalent histone modifications in controlling tissue development. For example, right here, and K4 trimethyl does not resolve the tissue types from here to there, but other histone mark can do. And so this is not about which histone mark does better job to identify different tissue types or cell types. It is about the biology, whether or not this histone modification actually controls the development of that particular tissue type, and where they do that job in the tissue. So again, the benchmark against the ENCODE reference data show very strong concordance. And if you want to really visualize this histone modification across the entire tissue, across all the pixels, you can perform unsupervised clustering to visualize all the different clusters. And now, as I say, you can directly visualize the different clusters in a spatial tissue context rather than just showing the beautiful UMAP. And so now you can see the 10 different clusters will resolve for the K4 trimethyl, and a very fine spatial resolution. You can sort of see, for example, right here in a brain, right between these 2 different layers of the brain, you can really resolve the subtle difference right here. Even in a heart chamber, it's a tiny heart, and it's an atrium chamber, it's a ventricle. You can resolve atrium versus ventricle, and even in the atrium, there's a hollow chamber. You can see there's a hollow chamber, and with sort of the blood cell or red blood cell clot in the middle. And so you can see all the different clusters using genome browser to look at on the peaks for different genes. You can also visualize those individual genes in a tissue. And so in this case, what you're looking at is the gene activity score. Since this is promoter activity histone modification, and but for other histone modification, you are again looking at either an activity or the suppression of gene expression. So basically, what I are saying here again, it's not just a gene expression, it's the biology, okay, the epigenetic biology associated with the tissue development and the cell type fate control. So similarly, if you integrate a single cell sequencing data, no matter single cell content tag or single cell RNA sequencing, turns out either one works so well. And once you integrate them together, and you basically move this two UMaps together, and look for the overlapping region for every single pixel here from our spatial content tag. And you know what are the major cell types in every single spot. And then you can go back to see where the different cell types are located. And again, in this case, what you see is whether or not this histone modification K4 method does play a role in the development of that particular cell type. All right. So what you are seeing is not just the gene expression or the presence of that cell type, but what you are seeing is whether or not this histone modification plays a role in that development of that cell type. All right, so you can also perform the shorter time analysis to examine this developmental process. For example, all the way from radioglia to excitatory neurons. In between, you have some post-mitotic premature neurons. And the similar shorter time analysis identify all the genes associated with this differentiation process. But what is most important is you can directly visualize this differentiation process in a tissue. Indeed, the radioglia cells are more in so lining around the ventricle once they become more committed to neuronal lineage, and in particular become excitatory neurons, and move away from the ventricles more towards the cortical layers. And we did something similar for the postnatal mouse brain, and in the similar region, we can resolve all those different regions of the mouse brain in the different layers of the cortical of the cortex, but also the corpus callosum and some sort of layers in the inner brain of this sort of subcortical layer. And so when you integrate the single cell data and the spatial content tag, you can transfer the cell types. All right, so here is so many different cell types. Once you see any kind of overlap between the spots, the pixels, and the cell types in the single cell reference data, you know, okay, that spot contains that particular cell type. You can go back to visualize where they are, right? So in my lab recently, we're very excited about aging and senescence. We're applying the single cell and the spatial ATAC and the content tag to investigate the immunosenescence in aging, but also the senescence in various diseases. So in particular, for the study of senescence in the context of epigenetics, so it turns out many different histone marks are implicated in the development of age associated with aging and senescence. I'm just showing the slide to show how sort of complicated this epigenetic mechanism is mediated by different histone marks. And so now we're working on sort of spatially resolving those different histone marks and how they do at a genome scale in human lymphoid tissues. I'm not showing any more data, it's still ongoing work, but very, very exciting to look into the aging biology and the stereosenescence. So since we're able to do spatial transcriptome and spatial epigenome, people constantly ask whether I can combine both, because the rationale is, all right, the epigenetic mechanisms control the gene expression, right? But can you actually measure the gene expression? Turns out it's totally durable. It's not too complicated, but I don't want to say that's trivial. And because these two chemistries are still quite different, we need to optimize quite a bit to identify the optimal condition that works for both transcriptome and the epigenome. So once that worked out, we have the data from both, for example, a tag and an RNA from the same spot, the same tissue pixel, and across the entire tissue region. If you just look at a tag, you can perform unsupervised clustering to identify all major tissue regions, and that works, and that worked quite well. You can also use the transcriptome to identify major tissue regions. That also works. And in this case, you can further compare so gene by gene and in terms of their accessibility and gene expression. I show you some of those. You do see pretty good, reasonably good concordance. And the integration with single cell reference data allows you to further identify all those different cell types. Now you can go back to see where they are. And very similar approach. But people may ask, you do global chromatin accessibility analysis to identify major tissue types or cell types, and you also use spatial transcriptome to identify major tissue and cell types. Which one can do a better job? And I would say that may not be a kind of perfect question. I don't think which one can do better or worse. Instead, I think each gives you a different message. And once you have both, you have much more sort of informative data set to really understand the biology. Right? And people may ask you what's the added value once you have this kind of co-profiling, spatial co-profiling. And so here I just show you one example. So when we have this co-profiling of the mouse embryo, we look at the differentiation from radioglia to post-mitotic premature neurons. And so in general, that makes perfect sense. I think from the ventricular layer, you see radioglia, and once they become more committed to neurons, they migrate away. But when you look at all the genes, I think the general hypothesis is the chromatin opened up first, and then you see the genes are expressed. And that's true in general for most of those genes, but there are some kind of outliers or exceptions. For example, those genes, and they have their chromatin opened up and remain open throughout the entire differentiation process towards the post-mitotic premature neuron. But an RNA, the gene expression is high in the beginning and very quickly diminished. And then when we look at what are those genes, my collaborator Gonzalo Castello-Branco immediately recognized those are his favorite genes associated with the oligodendrocyte lineage, not neuronal lineages at all. So this is interesting. That means, okay, in the epigenetically, those cells still maintain their potential to be oligodendrocyte lineage, even though they pretty much already decided they are gonna be neurons. They're gonna be the neuronal lineage cells. So this is an interesting finding, highlighting the importance of both. I think it's not about which one can do a better job. Once you have that, you understand better the biology. So we'll further perform this pseudo-time analysis for the epigenome data and the RNA. So when you look at the RNA, it does make sense. Okay, it's kind of monotonic transition from the radioglia to post-mitotic premature neurons. But when you look at epigenetics data, okay, this is a bifurcation. So a subset of those cells maintain the full commitment towards neurons, but a subset of those still maintain the epigenetic potential as oligodendrocyte or full oligodendrocyte lineages. And so now you can further resolve where are those different cells. So epigenetic states, they're all post-mitotic premature neurons, but there are two different epigenetic states within the post-mitotic premature neurons. All right, so we have done the spatial epigenome and spatial transcriptome co-profiling for cut and tag and RNA across all different histone marks and beautiful data. I don't have time to dive into the details, but interestingly, I think at least two of those samples are serial tissue sections. So we're now trying to do replicate experiment, but it turns out they are very much like a replicate experiments, and then you really appreciate how reproducible, from the different experiments, you can identify more or less the same tissue regions and pixel by pixel, very consistent results. And I'm probably gonna skip a couple of those slides to really highlight and what we are excited about. So the next step, but I want to emphasize what we have been done in my lab. So largely limited to what we're interested in. Okay, we're interested in brain, we're interested in the lympho tissue immunology and cancer, also derived from the hematological system. But I think Atlas Exomics has demonstrated the utility of the spatial epigenome sequencing across a very broad range of tissue types, and we haven't been doing, but you can see how sort of the broader applicable this approach for many different tissue types. So what we're excited about next in my laboratories, I know I'm talking about spatial epigenome sequencing. We're burning cache NGS every day, but I do appreciate the imaging-based spatial omics is truly powerful, right? Can we get a best out of both worlds? And so recently we have been conducting this high-plex immunofluorescence imaging of human lymph nodes and lymphoma using Codex, the Akoya Bioscience Codex platform. We apply this to human lymph nodes across different age groups. For example, here it's actually profile 12 different human lymph nodes across 4 different age groups. And we analyze only 5 of those. It's already close like 10 million single cells, and I feel we're gonna get over 20 million single cell from those lymph nodes. But what is super exciting is what was we mapped out using the imaging technology where the different cell types are. We can also identify where the senescent cells are. But in the same tissue, we're able to perform spatial transcriptome or spatial epigenome sequencing. And the spatial sequencing allows you to really explore the biology because the data is genome scale, right? But now you really can see exactly where we mapped out the different cells in the tissue corresponding to all different cell types because of the Codex, high-plex immunofluorescence Codex for cell typing. So now you can really kind of precisely pinpoint what cells contribute to that epigenetic state or transcriptional state and a truly single cell resolution. We're also excited about how we can further incorporate other layers of the omics, not just for fun, but actually really link the function to omics, no matter proteomics, transcriptomics, or epigenomics. I just show you one example, which is the metabolic function. And we are collaborating with Dr. Xie at UCSD to apply the super resolution Raman scattering microscopy to map the metabolic activity such as lipid unsaturation and glycolysis versus oxphos and so on and so forth. What you are seeing here is more like redox activity, so glycolysis versus oxidative phosphorylation. But every single cell you see here, we have the Codex data. We also have the spatial transcriptome or spatial epigenome sequencing data. So they can really link every single cell by cell, from the omics to the metabolic function. And so another interesting development in my lab, and I think Brian mentioned the Boston Epigenetics Society is excited about the genomic DNA-based epigenetic, but also epitranscriptomics. But nowadays, in the whole field of spatial transcriptomics and largely spatial transcriptomics is equal to spatial gene expression, but RNA molecules have extremely rich biology, very rich sort of life cycle, but we pretty much just look at gene expression. I feel that's biology-wise that's not that exciting. Can we really explore the rich RNA biology across the entire life cycle of every single RNA molecule, and whether I can actually do that in a clinical archival FFP tissue specimens. And so this illustrates the rich and intricate life cycle of every single RNA molecule, and we recently developed this technology called PathoDept. We already uploaded to bioRxiv, so if you're interested, you can check it out. But the key message here is, unlike conventional spatial transcriptome, no matter imaging-based or sequencing-based, you pretty much look at the presence of the messenger RNA, which is gene expression. But in our case, you can look at so many different things. You can look at the pre-RNA and a more mature RNA and the different splicing isoforms. You can look at large and small RNAs and the different microRNAs, and also you can kind of quantify the mutational landscape and like a single nucleotide variants, and so you can look at so many different aspects of the RNA biology directly in the tissue, and in particular in this case in the human archival FFP tissue specimens. Maybe just to show you kind of two examples. Okay, first of all, I want to emphasize, even though there's FFPE, but turns out the data quality is amazing, and we found that it can even surpass the data quality we collected from fresh frozen samples, and that's total surprise. But we found that that's actually true in many samples, and FFPE doesn't necessarily give you a lower quality of gene expression data, and actually, in many cases, RNA molecules are well preserved in the FFP tissue block. Just when you try to dissociate, you damage the molecules, the RNA molecules. But in our case, we do in-tissue chemistry. We don't have to dissociate, and we actually can probe a majority of those RNA molecules and obtain the data quality even better than fresh frozen. Okay, so one thing is we're able to identify not just the expression of the messenger RNA molecules, but also different splicing isoforms. For example, here, this particular gene, right? If you look at the expression, you do see expression sort of in the isocortex, in the hippocampal region, but when you look at the different isoforms, in particular, the skipping isoform is exclusively present in the dentate gyrus of the hippocampus. Okay, if you just do spatial gene expression, you would not see that at all. So now we are able to kind of look at many region-specific or cell type-specific isoform directly in the tissue and in the FFP tissue specimens. So another interesting aspect is we found that you can resolve so many different microRNAs and almost approaching the entire pool of the microRNAs in human tissue, and now you can you also have the gene expression. Now you can really understand the biology, how different microRNAs regulate their target genes, and you can visualize that directly in the tissue. As I mentioned, you can look at SNV, the single point mutations, to quantify something like a tumor mutational burden, right? When you count all the mutations, you can calculate the cumulative mutations across the whole tissue, and the high mutational burden region indeed correspond to the tumors. And so in this case, you don't need any protein markers. You can truly resolve where the tumor cells are. And furthermore, it can actually see different subclones or subtypes of the tumor based on the mutational landscape. All right. So can I use this slide to summarize what the future gonna look like? Okay. So recently we were able to use the AI driven algorithm to integrate on omics data with tissue histology to generate this super resolved single cell level spatial atlas of the transcriptome, epitranscriptome, and the epigenome across the entire tissue section, and potentially can integrate all different aspects of RNA biology and also the epigenetics into this tissue map. This information is very rich, way beyond gene expression or protein expression. So I think the key take-home message is not just about the technology we have and what we're working on in terms of biology we're excited about. The key take-home message is really together is better. I would like to thank all the people from my lab, but also so many collaborators across the whole community. I hope we will have more interaction with the Boston Epigenetics Society as well. And I would like to acknowledge the funding support and your attention. And thank you very much. If time permits, I'm happy to answer a few questions.
Thank you, Rong. That was a great talk. Our first question is going to come from Jason Wood. His question is, as the number of spatial omic technologies continue to grow, what do you see as the primary challenges on the data analysis side to help integrate and interpret all the data? And what directions or solutions do you foresee developing for overcoming these challenges?
Yeah, I think nowadays there are many different data types, and which means, okay, no matter what computational tools they develop, always better for this and maybe suboptimal for the other. So I'm not an expert in computational, but I have my own opinion. Very humble opinion. I don't want to convince everyone to agree with that. I feel one day all the different spatial mapping methods, it doesn't matter which better or worse. No, they are all great, and it turns out each technology gives you different information. They are complementary, they are synergistic, but project all the data to a single cell reference data. So that's my I feel that's a very good approach. In particular, single cell reference data being generated by multiple consortium, including Human Cell Atlas and NIH HeartMap and many other consortium. Gonna really build the reference data set, single cell reference data set across all major organs in human, in mouse. So that becomes the skeleton. And the spatial data, you just put your skeleton to you put your data to that skeleton. Eventually, that looks like a person, no longer just a skeleton. So that's kind of what I believe the best approach in the future. Maybe number two is how to do that, how to integrate that. I kind of believe in the generative AI and all those machine learning AI approach gonna truly transform how we do that in the future. That's just in the beginning.
So for our next question, do you have any thoughts on the value of DNA methylation information as an additional layer to this work? And that comes from Brad Langhorst.
Yeah, that's actually exactly what I suspect what happens to the FOXP3, because FOXP3 locus and remains open in many different cells, many different T cells, not necessarily regulatory T cells. That's what we saw. But what really initiates the sort of the transition to regulatory T cells, that's the DNA methylation of that specific TF locus. which we don't know yet, but it is totally possible to do spatial methylome sequencing and even combine spatial methylome with spatial ATAC. Totally possible. We're not working on that, but I believe people in the field, yeah, that they might get there one day.
Our next question is from Ankush Sharma at Neosome. How does patho-DBIT compare to other spatial transcriptomic techniques, particularly in its ability to analyze FFPE tissues?
I kind of already mentioned that kind of comparison. I think if you're just looking at gene expression, I was totally blown away. Actually, this is as good as the probe-based spatial transcriptome, right? So I thought it's no way to do better than probe-based, but turns out we get almost the same data quality, sometimes even better. But think about, since this is now probe-based, this is now targeted, you can look at so much more than gene expression. So yeah, that's what I can tell you. And I was totally surprised that data quality is that good. Yeah.
Our next question's from Mayor Doak. Thank you. Very nice talk, Dr. Fan. I have a question about linking single cell omics to metabolomic activity, dynamics, and function. Is it possible to detect calcium signaling activity in the pancreatic beta cells using single cell omics?
Oh, so I'm not so familiar with this, but I think calcium signaling, you need to do live cell imaging, right? It is totally possible to do live cell or live animal imaging of the calcium signaling. But afterwards, you fix the tissue and you perform spatial omics, and then you can kind of link the live cell tracking data to the omics. And I think people are working on that.
So I know we need to wrap up soon. So the next questions we could send to you that you could reply back to people via email. But thank you so much for this talk. It was, riveting, and I think people really enjoyed it. We'll forward the rest of the questions to you so that you can make it to your next meeting. Thank you, Rong.
Thank you, John. Yeah, thank you for having me.
Yeah, we look forward to seeing you at the next Boston Epigenetics Society event.
Yeah, looking forward.
All right, thank you.